Well, and thank all of you for coming out this afternoon. I'm excited to tell you about bacterial genetics because I am always excited about bacterial genetics and also how we can apply bacterial genetics to biofuel production. So just to kind of start out and make sure everybody is on the same page, have some learning goals for the lesson. So the first of which is to understand the basics about genetic approaches in bacteria. Once you know these basics, you can apply genetics to nearly any problem. The second feature is how CRISPR works, because we're going to be applying CRISPR technology to some of these problems, and understand specifically how we're going to use CRISPR to understand features of organisms that are important for bioenergy. All right, so the first thing I want to tell you about, maybe you don't have a background in bacterial genetics, so what is bacterial genetics, and kind of how do people think if they're considered bacterial geneticists? And I like to think of bacterial genetics this way. We break things and see what happens. So this might be a little different than a lot of other disciplines that you might have thought of, where you think about building stuff up. You think, oh, well, if I want to understand that thing, I'm going to be able to put all the parts together and build it up. In a lot of the cases with bacteria and with other organisms, the organisms themselves are too complicated to build from scratch, at least for now. But we can break individual components of the organism and ask, well, now, what was that piece of the organism doing? Why was that important to the organism? And that's going to come in handy when we think about bioenergy as well. So I want to move on to kind of like the central dogma of biology. Now, I think probably the vast majority of you know this or have at least some familiarity with it. And I'm not bringing this up just to try to teach you what the central dogma is, but rather I want to point out something really important, which is, of course, when we have DNA, we have a gene that's transcribed into RNA and then turns into protein. Any changes that we make to the DNA, right, go through the process of transcription as well, and then end up in the final protein, right? So these final proteins are, in many cases, are going to determine what the output of that cell is. So if we make changes here, that's going to change the protein. If we somehow have a way of changing the way the RNA is expressed, maybe we have more RNAs or less RNAs, we end up with more or less protein, right? So all of this is one process. I don't want you to think about it kind of like as a textbook sort of thing, but rather if we break individual parts of this, we end up breaking the final product as well, and we change the behavior of the bacterial strain. All right, so what does this kind of look like actually in bacterial cells? So what we want to do, right, is we want to change this DNA, and then we want to look for changes in function. So what am I showing you here? I'm showing you some bacteria on a plate. So what do you think? Do you think that these two bacteria look similar or different? I'm asking the audience. Help me out here. Do they look similar or different? They look different. They're different colors, right? Well, so they look pretty strikingly different, right? This one's like a hot pink, and this one's kind of like more of an orangey color. But it turns out the whole difference between these two bacteria is just one gene. There's only one gene that's actually different. So one little piece of the whole DNA for this whole bacteria is different, and that gives you these drastically different outcomes, right? And what is this actually? So the genotype, the DNA, this strain is LAC-Z+. So this strain has a gene that will take the lactose sugar in the plate, eat it, and then convert it to acid. And that acid is actually what you see as this pink color. So that's what we call LAC+, because it can eat the lactose. Now, for this strain, we have a deletion of that LAC-Z gene. Just one gene in this bacterium totally changes the way the bacterium looks, and it gives you this lactose minus phenotype. So this bacterium can eat lactose, and this bacterium can't, and the only difference is one gene. So bacterial genetics is a very powerful approach to really change the behavior of bacteria just by making really simple changes to one place on the DNA. All right? I don't expect you guys to memorize this kind of nomenclature here, but just so you know that the individual genes that I talk about are often in italics. And so that's what I'm referring to when I say an individual gene. It's a gene that will make an mRNA that will encode a protein that will have some activity. In this case, that activity is eating lactose. All right, so we want to change the DNA to look for changes in function. So how do we do that? Well, one of the ways is just to precisely remove a single gene. And I'm going to show you kind of how that works in general. I should just mention that some of the figures that I have are a little bit complicated, but just follow me and we'll walk through them. I don't know why, but for whatever reason, even really simple procedures have really complicated pictures on the internet. And I don't know, I guess it's to make people feel smart, but you're going to be so much smarter after I explain this because you're going to know exactly what's going on. And you'll just look at this figure and roll your eyes because you'll be like, that's way too complicated. All right, so how do we delete genes? It turns out we just replace them with another gene that doesn't do that thing. One of the most common ways we do this is actually by taking an antibiotic resistance marker. So this is a kanamycin resistant cassette. What that means is this is a gene that if you put it in that bacterium, will make the bacterium resistant to kanamycin. Okay, that's cool, right? And we can actually select for bacteria that become resistant to kanamycin. So bacteria that get this DNA become resistant to kanamycin. Awesome. Now, let's say here, I've got a yeast orf. It's exactly the same kind of scenario in bacteria. Let's say that this is that LAC-Z gene that we're replacing. We simply take that kanamycin resistant cassette, put it in the place of that LAC-Z gene. Now the LAC-Z gene is gone, and the kanamycin gene replaced it. So that particular cell would have a kanamycin resistant phenotype that's new. It's now resistant to kanamycin, but it's no longer producing LAC-Z, so it can no longer turn red. And we target this kanamycin cassette into the genome at a specific gene by just having pieces of DNA that look like the genome, and that's called homology, right? So these pieces of DNA that are just outside of the kanamycin marker look like the genome, and that causes that piece of kanamycin DNA to go in and replace the existing DNA. It's kind of shown here at the end. So what deletions are precise gene replacements. Now, let me think about this for a second. In that first picture, I knew what genes were involved in consumption of lactose, right? Because other people have done studies to show me what those genes are. But what if I didn't know? Right? There are more than 4,000 genes in most bacteria. We don't know what they all do, right? Even in really well-studied bacteria, we don't know what about a fourth of the genes do. So how would we figure it out? Well, we could just take this kind of approach and actually just systematically delete every single one of those genes, so all 4,000 of them, which turns out is a lot of work. So we call these things deletion libraries, and these things are really awesome because what we have is a deletion of every single gene exactly lined up. In many cases, like for this organism Bacillus subtilis, we've made a library that has 4,245 deletions in it, and we're keeping these things arrayed in these little plates with these little wells. So every single well is a different deletion. Now, if we didn't know, for instance, what gene was involved in consuming lactose, what we could do is just take all of these strains and put them on lactose and then individually check to see which one didn't turn red. That's how we could do it, right? But that would be a lot of work. We'd have to make 4,000 plus strains just to figure that out. So that's really painful. But these are really nice libraries. Now, unfortunately, there are some genes of bacteria that are just too important to get rid of, right? Some genes, remember I showed you that flow chart that had DNA, RNA, and protein? Usually genes involved in that process of turning the DNA into RNA, or copying the DNA, turning the DNA into RNA, and then turning that into protein are super important. Those kind of genes are called essential genes. You can't get rid of them, right? You can't delete them. And so when you try to delete them, you actually don't succeed. So these libraries can include deletions of essential genes. There's no way to study them using this approach. So this is a great approach for finding a lot of different phenotypes, which is those different outcomes for the bacteria on the plate. But they're not a very good way of studying the genes that are absolutely the most important in bacteria. And I'll get to a way that is how we study those. So building these libraries is awesome, but it's hard to make them. So what would be easier? Well, how about something called transposon mutagenesis? So transposon is a protein that takes its own little piece of DNA with it, like that kanamycin marker I showed you on the previous slide. You can transform it into bacteria, and it will jump into the DNA randomly. So it will go and disrupt a gene at random, and you end up with this plate covered in bacteria. But a lot of these individual colonies, they have a disruption of a gene. They might not have a disruption of a gene, but there's some way changed from the parent organism in that they got this transposon stuck in them. So this is a really great way of disrupting genes that doesn't take a lot of effort, and we can do it as a big mess of strains, right? We can take this transposon and dump it into cells thousands at a time, disrupt many, many genes. But now we don't know, out of the box, like on the plate, whether or not that individual colony has a mutation in a gene that we're interested in. We have to then screen this plate for potentially like differences in color, like I showed you at the very beginning. So transposons are really easy to make libraries from, but they randomly transpose. So how can we learn about what's going on in the cell using this easy method of transposition without actually having to go through a lot of effort? So one of the ways to do that is by TNC, and I apologize for this horrible slide. It has way, way too many things on it. Just pay attention to what I point to and follow along, and it will be much less painful. So the first step is just like the last step that I showed you in the previous slide, right? We have to make those strains. The way we do that is we just take our transposon and we put it in with our transposase into the bacterial cell. Okay. Now we have a lot of different strains because that transposon went into DNA all over the place, disrupted all kinds of different genes. That's what's being depicted here. Now what we can do is we have a big mess of cells that have transposons in them. That means it's a mix of a whole bunch of genes that have been disrupted. We can treat that with whatever condition we want. Let's say for GLBRC purposes, that would be some sort of biofuel production condition. And I'll get more into the specifics of that later on in the talk. So now we have our initial population. We have a population that's been put through some kind of stress, and we have our final population. So some of the genes that were needed for that stress are now gone. And we can figure out what those genes are simply by sequencing where the transposons ended up in the strain, which is all this mess down here. You don't need to know the details about that. You just need to know that we can, in fact, sequence these things and figure out where the transposons went, which means we can figure out what genes were disrupted and which genes are important for that particular condition. So what does this look like? This kind of data look like? Well, I'll show you here. Each one of these red bars is a transposon insertion into a genome. And what you can see is here, this blue gene here and this blue gene over here have a lot of insertions in them. So what does that mean? It means that you can very easily disrupt these genes with a transposon. Nothing happens in that condition. Those genes aren't needed. You can break them and break them and break them. Nobody cares. Cells don't care. Well, what about these genes in red, right? They don't have these transposons inserted into them. These genes are super important. If you remember earlier, I told you about those deletion libraries and how there are some genes you absolutely cannot delete. That's what these genes are. These are essential genes. So these genes are all involved in actually, when you have one cell and it's dividing into two cells, it's involved in making that little septum that pushes the cells apart there, pushes them into the middle and then pushes them apart. So these are absolutely important. Without this, you can't start with one bacterium cell and end up with two. That's a huge problem if you're a bacterium, because when you're a bacterium, all you want to do is make two cells. I mean, that's like the goal in life. That's really it. So these genes are critically important for that process. They're absolutely essential. Can't delete them. And that's what this data tells you. If you can imagine, so this was just done under the ideal growing conditions, but you can imagine that there are genes that are important for a particular condition that might also become important, and then you wouldn't be able to delete them. For instance, I showed you at the beginning those cells that are growing on lactose. If the only source of energy was lactose in that plate, the lac genes would also become essential. right? So it depends on the context whether or not a gene is essential. That's the point I want to make, and I will get back to that point later. And so finally, I want to just talk about one other general strategy for looking at the relationship between genes and their phenotypes, and that's an evolution strategy. So typically the way this works is you have some diverse strains, or maybe you don't, maybe you just have this one strain. And what you will do is you'll actually grow the cells up under a condition that is of interest to you. So let's say you're trying to identify genes involved in ethanol tolerance for a bacterium. You would grow those cells in medium that also contains ethanol, and then only the best growing cells would grow up. You would grow them again, and you would just keep enriching for that best growing pool. When you're done, you'll have these bacteria that have mutations in them, and you'll be able to map where those mutations end up. So you'll be able to figure out which genes got mutations. So this is a process that allows you to enrich for genes that have the right phenotypes, the right outcome that you're looking for. So earlier in the talk, I talked about ways of mutating genes. So we can make a precise deletion, or we can use a transposon that jumps into the gene and disrupts it. Does anybody know where the mutations come from in this process? Right, there are mutations here that we selected at the end that grow better. Anyone have an idea? It's okay. So it turns out that as the cells are growing, they just can't copy their DNA perfectly, right? So at some low rate, low frequency, cells will actually get mutations in particular genes. Now, most of the time, that leads to the cell being crippled, so that just falls out of the population of cells. But in some cases, these cells actually grow better because of that mutation, and those are the things that you enrich in this process. So, up until this point of the talk, I've been saying things that are pretty hypothetical, right, or very high level, like these are genes and we need to figure out what they're doing. These are ways that we figure it out. So how do we actually apply this kind of approach for bioenergy purposes? So one of the GLBRC has been around now for more than a decade, and I'm happy to say that I participated a little bit at the very inception of the GLBRC, and now I'm around again, which is pretty fun. And one of the things that we did at the very beginning of the GLBRC was to try to take a strain that we knew very well, was very dear to us, E. coli, and make it produce ethanol. Now, you might be thinking, oh, E. coli, isn't that the thing that makes you sick? Well, yeah, there are some E. colis that make you sick, but the E. coli that we were using was a very docile E. coli. It's the one that's around the lab. It doesn't cause any harm to anyone. But it has a lot of really great genetic tools, and we've been using it in the laboratory for a long time. So we thought, oh, maybe we'll be able to use this strain to make ethanol. And so what we did was we did this exact kind of evolution experiment. So here is the experiment. We took E. coli. This is called wild type. Why is it called wild type? Well, it's the natural strain that we make comparisons to. Now, of course, it's been in the lab for decades, so it's not really natural anymore. But what wild type means is this is the strain we're starting with. We're comparing it to this genotype. Now, what did we do? We grew it in increasing concentrations of ethanol until we got this strain out, MTA156. So MTA156 is quite a bit better at growing in ethanol than the wild type strain. So what I'm showing you down here is just the growth of the bacterial strains. The wild type strain, or the starting strain, the lab strain, is in blue, and you can see it kind of growing up here happily. Then we add ethanol, and then it's like, oh, why? Why did you do this to me? I'm just a lab strain of E. coli. I just wanted to be happy and make more E. coli. But if you look at MTA156, you'll see it growing happily. We add ethanol, and it's like, oh, okay. And then it's like, great. I'm going to get through this. I made it. Awesome. So we evolved a strain of E. coli, which is not naturally resistant to ethanol, that is now resistant to ethanol. Now, here's the kicker. So we did this evolution experiment, and the strain, of course, has mutations in it. Remember, I said those mutations are the things that we're selecting for. But the strain will have mutations that are also unrelated to the phenotype too. One thing that we had to do was try to reconstruct the difference between this green strain and this blue strain. And we've ended up finding that it took 3 separate mutations of 3 separate genes to get us to the point where we can even recover a little bit of that growth. So these evolution experiments are really nice. We can take advantage of a natural process to generate mutations in genes that give us these phenotypes that we want, better growth in ethanol. But they're really complicated because we get a lot of mutations back out, and we don't know exactly what genes are important because we have to reconstruct the strains to figure that out. We have to move these mutations into a new strain and see if that new strain has the same properties. Does that make sense? Now, the new center is no longer involved in using E. coli to produce ethanol. That's actually old news. The new center is trying to use exciting new strains like Zymomonas mobilis, which is already naturally resistant to ethanol, to produce things like isobutanol, which is more like jet fuel. But I'll get to that momentarily. So now, just to review, we talked about several different strategies for looking at how genes function, genetics. So these single gene deletions, but the limitation here is we don't know what gene's important on the onset. So if we go and we start deleting individual genes, we'd have to get real lucky because there's 4,000 of them, right? So if we don't have a way of looking for that, or we don't know from some other studies which genes to delete, we could be deleting things for a long time. Well, one option there would be just to delete every single gene, but of course that's a real pain. It took my friend like 4 years to make that deletion library of Bacillus subtilis, and I think he suffered a lot. One of our favorite memories of him was just him sighing deeply when he came in to make that library every day. Just, oh, so that's very onerous, and I can tell you that for sure. Another thing that you might not consider, but is definitely important for labs, is if once you've made that library of 4,000 things, you got to keep it. So you got to keep it in your freezer, and you got to keep it separated so that the strains don't all mush together, because if they mush together, then you got to figure out how to get them back apart. It's a big pain. It's also painful to screen because every single well is different, right? So every time you do an experiment, you have to carry all those wells through individually. So very challenging. Transposon immunogenesis is great because it's super easy. Just get the transposon, transpose it into your genome, knock out a gene. Awesome. But it's random. So you have to figure out from that random pool, how are you going to get the genes that you're interested in? And the same is true with transposon sequencing as well. Even though you can look at all of the genes at once, you have to have a clever way of finding conditions where those genes will grow differently. And finally, with evolution, it's great because you can have all sorts of different kinds of mutations, but in the end, when you have a lot of mutations, you don't know which ones are causative. Now, all of these techniques are awesome, and they're kind of like the fundamental backbone of genetics, but they have a major, major limitation. And that major limitation is that none of these strategies will allow you to target those essential genes that I was telling you about. Now, you might think, well, doesn't evolution allow you to get at essential genes? And it does, but it doesn't allow you to target them, right? I can't target my evolution to a particular gene. I just let the strain evolve and I see what mutations come out of it. No way to target it with any of these other approaches, right? If you try to delete the essential gene, the cell is dead, you don't learn anything. Transpose something into that gene, cell is dead, you don't learn anything. So we can't target essential genes with these approaches. So what are we gonna do? Well, luckily there's a new technique out called CRISPR that allows us to do this. Now, when I say CRISPR, what do you think? What's the first thing that comes to mind? I'm not letting you go on this one. Somebody's gonna tell me something.
Cas9.
Cas9, great, yeah, great. What about uses of CRISPR? What do you hear? What do you hear about for uses of CRISPR? What do you hear about in the news? You heard anything? Gene editing, gene therapy, great, yeah.
Designer babies.
Designer babies, yeah. Creepy, don't, okay. My talk is not about designer babies. Anyway, but yeah, gene editing and maybe even human gene editing, right? So changing DNA in a specific way to have a specific outcome. But what if I told you that that's not even the most interesting use of CRISPR? Ha, stay tuned. But before I get really deep into how we're using CRISPR, I thought it might be appropriate, since this is a class with a kind of a broad group of people interested in it, give you a little bit of background on CRISPR. What is CRISPR and why did it come about, and how is it so powerful? So, discovery of CRISPR. Well, it turns out that the discovery of CRISPR was pretty mundane. Back in the 1980s, people were doing what people did in the 1980s, which was to sequence a single gene. So sequencing a single gene was really hard in the 80s. People would spend years of their life just sequencing one gene. And they were super pumped about that because they did not know the sequence of the gene beforehand, right? So they had no idea what their protein would look like in terms of amino acids or anything, because they just didn't know the sequence. Turns out that this Japanese group, though, they had sequenced their favorite gene, and they found something really weird at the end of it. They found this repeated sequence followed by some not repeated sequence. And they said, okay, well, this is a repeated sequence, and this is just the space between that repeated sequence. Biological significance? Unknown. That's the discovery of CRISPR. There's a weird repeated sequence and some not repeated sequence, and we have no idea what's going on. Cool, see ya. But it turned out that around 2002, people started sequencing a lot of bacterial genomes, and they saw these things all over the place. Well, what are these things? Well, not knowing what they did never stopped anybody from naming something, right? So what they named them were CRISPRs, clustered regularly interspaced short palindromic repeats. Yeah, that's what that is. How many people knew that that's what CRISPR stood for? Oh, we got a couple. But yeah, I mean, the acronym doesn't really describe too much about what CRISPR really does. So, but you know, we were learning. So people still didn't know what they did. They saw them, but they had no idea what they did. Up until something really exciting happened. Now, before I tell you about that exciting thing, I want to tell you about phages. So phages are viruses that infect bacteria. Well, it turns out that 5 to 10 phages, there's 5 to 10 phages for every bacterial cell on the planet, which means that there are 10 to the 23rd phage infections per second. I don't even know how to think about that number. That is such a huge number. It's crazy. I mean, what do you compare it to, right? There's nothing really at that magnitude that you can even consider. So it's a crazy huge number. So how do bacteria defend themselves? Oh, by the way, this is a bacterial cell. And these are the actual phages infecting it, which I think is a super cool picture. And in some cases, you can actually see the phages injecting their DNA into the bacterial cell. So the phages are basically going to take over the bacteria by infecting it. So it's a very interesting process. So how do bacteria keep this from happening, right? There's so many phages. Well, it turns out that they use CRISPR for that. And the way that we figured that out was also, not obvious. So it turns out that there's a yogurt company outside Madison called Danisco. And if you know anything about yogurt, you know that they have to grow a lot of bacterial cultures to ferment their yogurt, right? So what was happening? Well, they grow up a big culture of bacteria. Things were awesome. They're making yogurt. And then all of a sudden, all the bacteria are dead. Well, that stinks, right? How did they die? Phage. Bacteria viruses, right? Went in and destroyed all the bacteria. Well, that is such a bummer when you're trying to make yogurt. So there are scientists there that were just saying, okay, what if we had a bacterium that was resistant to phage? And remember, I showed you earlier how you could do evolution experiments to get mutations that allow you to become resistant. So that's what they did. They did a very simple experiment. They took their bacteria, they mixed it with phage, they had a whole bunch of that bacteria, and then they looked to see what bacteria survived. Now, the vast majority of the bacteria died, but there were a few bacteria that survived. And what they found was very surprising. They found that pieces of that phage DNA actually got put into those spacers in the CRISPR, right? Those boring things that didn't have any relationship to each other, that were just in between the repeated sequences, actually had pieces of the phage in them. And so what you can see here is, this is the normal CRISPR spacer in this particular strain, and it has a lot of sensitivity to this particular phage, this 858 phage. Now, when they took a resistant colony, what they'd seen is that it had actually acquired 2 new spacers. And those spacers matched this 858 phage. And what they found was that there was a massive drop in sensitivity, multiple orders of magnitude drop in sensitivity to this phage. Now, the really cool thing was that they took these spacer sequences and they tried to use them against a different phage. Didn't work, right? So these spacers were specific to that particular phage. That was the answer. And they could take these spacers and move them into another strain that had never seen the phage before, and it would be resistant. So this was the demonstration, right? That CRISPR was targeting phages, and actually by incorporating some piece of the phage DNA. So how does this process work? Well, this is our bacterial cell here, this box. We have our friend the phage. Again, it's injecting its DNA into the cell. I want you to follow this red piece of the DNA here in particular. Here in the bacterial cell, we have a bunch of genes called Cas genes, CRISPR-associated genes. So these genes are actually going to interact with this piece of the phage DNA and stick that DNA into this CRISPR array, into the spacer. So now the bacteria is armed against attack by this phage. How does it work? Well, the bacteria transcribes this CRISPR array. Remember, we go from DNA to RNA, and we make these RNAs called guide RNAs, gRNAs. And these guide RNAs, it turns out, absolutely match that red sequence for the DNA. So this is that Cas9 you were talking about earlier. The Cas9 then recognizes that piece of DNA using the RNA that comes from the CRISPR array. So the RNA matches the phage DNA. That phage DNA is degraded. So what's the coolest thing about CRISPR? It's not that it can edit. It's not that it even cuts up DNA. It's that you can take a protein and direct it to any piece of DNA you want just based on this RNA sequence. And this RNA sequence is only 20 nucleotides. What does that mean? That means I can go onto the internet right now and order a whole bunch of 20 nucleotide pieces of DNA. They'll be here tomorrow, right? No problem. I can make as many of these things as I want. I can target as many genes as I want. No problem. And that's exactly what I'm going to do to improve our biofuel relevant strains that we have here in the center. So this is CRISPR. This is CRISPR cutting. But what if I told you that that wasn't the most exciting thing about CRISPR? I promised you earlier that it wasn't. And in fact, I have an article from The Atlantic here that says exactly the same thing. CRISPR's most exciting things have nothing to do with gene editing. Look, and we even broke the scissors. We don't care. We smashed it. So what can you do with the broken scissors? I mean, you can't cut paper with it, right? That's kind of lame. So what does a broken scissors do? Well, let me tell you something about CRISPR interference. This is the broken scissors, and it's very, very powerful broken scissors. So we have this Cas9 protein. Now remember, Cas9 cuts the DNA. But what we've done is we've actually made mutations in that protein. Well, we made mutations in the DNA that encodes that protein to make this inactive version of Cas9. So this version of Cas9, it'll be guided to a particular piece of DNA by this guide RNA. And again, we just need to have this sequence that matches the DNA anywhere to make that happen. So now we have a dCas9 guide RNA. And once it gets the DNA, it doesn't cut. It just sits there like a brick. It does nothing. It does absolutely nothing. It just sits there. But it turns out that doing nothing is awesome in this case because it completely blocks the enzyme that makes RNA from DNA, right? Remember when I showed you that central dogma slide at the very beginning? We had DNA going to RNA. dCas9 is blocking that process. It's preventing RNA polymerase from doing its job. It's allowing us to turn down that gene. So keep in mind that genes aren't just on all the time. They're only on when RNA polymerase comes and turns them on. So what this is basically is a dimmer switch for genes, dCas9. Does not cut, doesn't need to. So what are the cool things about this approach? Well, again, as I mentioned earlier, this 20 nucleotide sequence is all we need to change the target on DNA. No problem. we can actually just make multiple of these guide RNAs of the cell and target multiple genes, because in some cases, you knock out one gene, nothing happens. It takes 2 genes to knock down to have any kind of outcome, right? You can imagine a scenario like from the beginning of my talk where we had that lac gene, right, that ate the lactose. If we had 2 copies of that same gene and we knocked out one of them, we would still be able to eat the lactose. Only if we had knocked out both copies would we have a chance of figuring out what that gene did. Also, we can turn on the knockdown, which sounds kind of weird because we're knocking something down, but we can turn it up and it goes down. Anyway, that's a little weird, but trust us, we can do it, and it's really awesome. So back to the main story here, the essential genes. Remember, I told you none of those techniques could go after essential genes, and these essential genes are super important, right? They're the things that encode the ribosome, which makes the protein, right? They encode the RNA polymerase, the DNA polymerase, the things that make the fundamental parts of the cell. But it's really hard to deal with them. So here's a bacterial cell. Each one of these red dots is an essential gene product. Now, remember I told you that if we try to delete those products, the cells just end up dead. So we didn't learn anything about essential genes other than that they're essential. What if I told you that we could turn down that dimmer switch, make fewer essential gene products? Well, these cells are actually viable. They can actually live, even though they have less essential product. As long as they have essential product, they can still live. But it turns out they're sensitive to perturbations. Now, what I'm going to show you is about a drug, but it could easily extend it to any kind of bioenergy-related stresses, and I'll show you that momentarily. So here, what I've done is I've taken that partial knockdown cell, and I've given it a sub-inhibitory concentration of an antibiotic. It's a little complicated, but bear with me. So we're targeting 2 different places on the cell. The cell can still live, but if we target that partial knockdown with an antibiotic that also targets that same part of the cell, now there's not enough of these red dots, right? There's not enough essential protein function in the cell. That cell is dead. So we can actually figure out what that drug is doing. We can figure out what stresses are in plant hydrolysate. We can figure out what stresses are caused by isobutanol being present in the culture. We can figure out anything like that. That's how powerful these techniques are. So how do we generate these knockdown cells? Well, what I'm showing you here is just a toy version of this where we're using CRISPR, Cas9, to knock down expression of a protein that's just red. So we can see it because it's red. Now, if we turn on the system, which remember paradoxically turns down the protein, we can see the amount of red protein going down. And we actually have really nice control of this, right? Here's our level of inducer. More inducer, the more control we have. But you might have also noticed that even without turning the system fully on, we have a little bit of a knockdown anyway. And it turns out that that's a great amount to make these kind of partial knockdown cells, these viable cells that are sensitized but not dead. So in a previous example I'm showing you here, we made knockdown library of all the essential genes in a model organism, Bacillus solus. So Bacillus solus is a lot like E. coli, except it's evolutionarily diverged, but it's an organism that we keep around the lab to study. Every single one of these dots is actually a separate knockdown strain, except the outside rows and columns, which are wild type. Remember, they're the happy, friendly strain that hasn't been perturbed. And you'll notice that there's not a lot of difference, really, between the sensitized cells and these unperturbed cells. That is until you treat them with some sort of inhibitor. In this case, the inhibitor that we're using is an antibiotic. And you can figure out exactly what gene is targeted by that antibiotic, because that particular colony is gone. It's dead. So there's a synergy between the loss of that essential gene product and this particular antibiotic. But again, remember, it doesn't have to be an antibiotic. It can be any stress the bacteria encounters. And you can see other cells are sensitized as well. These aren't the direct target, but these are related genes. So they're involved in making a related product in the cell that this gene is involved in as well. And so I should mention that this isn't the only thing, of course, that we can do with CRISPR-I and essential genes. We've been able to do a number of things. I unfortunately don't have time to talk about them all today, but That was in a case where we were able to figure out how an antibiotic works. We could figure out how bacteria survive in any condition using this sort of approach. We can figure out how genes are kind of connected in these complex networks, because remember, we've got more than 4,000 different genes in these cells, and they all work together in complicated ways, and we can figure that sort of thing out. We can understand the growth of the cells. This kind of thing is very important if you're thinking about growing cells up in a fermenter and looking at how they grow under different stress conditions with different genes. So now I want to focus on something that's a little bit closer into the bioenergy center, which is that most bacteria that we have aren't E. coli and B. subtilis, right? Those are the ones that we've kept around the lab for forever. They're the happy lab strains. Most of these bacteria, I'm showing you all around here, you can't read it, that's okay. They're no tools. We can't do simple experiments in those organisms. But this is another situation where CRISPR comes to the rescue, right? So CRISPR is generally useful in a lot of these sorts of cells, and the reason why it can be useful is because we put a lot of effort into building tools to transfer CRISPR into these other organisms, including right in here, which is one of the strains that we like to use in the center, Zymomonas mobilis. So how did we do that? Well, we built this modular CRISPR-I system. So it has all the parts that I introduced earlier to you. It has the dCas9 gene. It has libraries of guide RNAs so that we can target many different genes. So it's all in this particular piece of DNA. Now, we can actually insert that piece of DNA into some kind of fancy plasmids that will go into a bacterial genome and then mate them. Yeah, maybe you maybe not have thought that bacteria mate with each other. They do. They mate and they transfer DNA. They actually have pili that will insert into the recipient bacteria and transfer that genetic material. So all we need to do to transfer CRISPR-I is to mush the strains together. That's it. Very nice, easy system to use. And then finally, the CRISPR-I system integrates into the chromosomal DNA. That's awesome, right? Why is that so awesome? Well, we need the CRISPR machinery to be part of the bacterial genome. If the CRISPR machinery is floating around as a piece of DNA that's outside of the genome, we'll have to put pressure on the bacteria to keep it. And the way we typically do that is by adding an antibiotic. Now, if you can think of like a big batch of bacteria, like a giant fermenter full of bacteria, we're not going to want to add antibiotic to that, right? That's going to be a big mess. It's going to be a big expense. Having it integrated in the genome means that it will be stably maintained just like the rest of the genome, and the bacteria will just continue to produce that piece of DNA. So that's super important. Now, how well does this system work? I'm going to show you a variety of bacteria, none of which are bioenergy related, but I promise, I will show you one in the end. So here's mobile CRISPR-I function in a bunch of different bacteria. What I'm showing you again here is knocking down that red protein. We can see that red protein. We can tell when it's knocked down. And you can see a variety of species on this axis. Many of these are actually pathogenic bacteria. Remember, I'm in the School of Pharmacy, so there's a lot of interest in finding new ways to beat up pathogenic bacteria. But you might say, well, okay, we know about these pathogenic bacteria. We've been studying them for a while. But what if you took some kind of like completely outside the box bacteria? Would you be able to transfer your CRISPR-I system into it and be able to turn down a gene in it? Yes, the answer is yes. Let's say we took a bacterium from the top of a piece of cheese, which we had, luckily, because we collaborated with Rachel Dutton's group, who studies the cheese microbiome. We got this bacterium off a piece of cheese, and you can see that we have a significant reduction in the red protein when we use mobile CRISPR-I in that strain. So we can control genes in a lot of weird bacteria. Bacteria people have never used in the lab for anything, right? So it's a very widespread approach that can be used on many, many different types of bacteria. And that's really important for the Bioenergy Center because we never know when we're going to get a new species of bacterium that could be really valuable for producing biofuels. And we need to be able to go into that species immediately and understand what the genes do. And so mobile CRISPR-I allows that sort of thing. So now, finally, to bioenergy. I promised the whole talk wasn't a bait and switch. here is kind of the simplified version of what we're doing in the Bioenergy Center. So we start with plant material. This is a picture of switchgrass. This is one of the types of plant material that we like to use. And we like to use that material because it grows well on land that is not optimal for growing other crops. We don't want to have to compete with crops that people use for food. We want to use land that is otherwise not very useful for that purpose. Now, we take this plant material, and through the magic of engineering, which I'm sure other people could explain better than I, we get sugars from that plant material. And these sugars we take and we feed it to microbes. One of those microbes is Zymomonas mobilis, and out of those microbes, ideally comes valuable products. This is isobutanol, for instance, jet fuel. We have some other people in the center that are interested in making other fancy lipids that might be useful for various other things, including other types of fuel. So this is the very simplified, high-level view of things, right? But in reality, it's a lot more complicated process and it involves a lot more nuts and bolts and gears. So here's the actual process. Wow, there's a lot of stuff going on here, right? There's a lot of different variations in the way the crops are grown, right? Just from year to year. You can imagine one year you get more rain, one year you get less rain. You've got a lot of different kinds of crops. You've got a lot of different ways of getting that sugar out of those crops once you get them. And then you have a lot of different kinds of sugars and other things that you can process. You have different organisms you're dealing with. So bacterium, right? Bacterium. So a lot of different kinds of bacteria here, a lot of different species, like very different relationships evolutionarily, and a lot of different kinds of products to get out of here. And one of my favorite representations actually of the type of problem that we're dealing with in this scenario, we're getting all kinds of stuff out of these plants. And of course we wrote sugar there because that's what the bacteria like to use. But this is the picture from my scientist about the sugar. So there's sugar, but it's in this globby goo thing, you know? This kind of mix of plant goo that came out, and that goo has the sugar in it that we want. But it also has all kinds of other stuff. It has acids that are hard for the bacteria to deal with. It's got all sorts of other inhibitors of bacteria growth. We don't even know what they are, right? We're pushing really hard to figure out what they are, but we don't know right now. So how do bacteria survive long enough in this condition to end up producing the isobutanol? And that's what we're trying to figure out. We're trying to figure out what genes are important in the bacteria. And of course, some of the genes that are going to be most important for growth in these conditions are those essential genes, right? And without any other approaches other than CRISPR, we're not going to be able to systematically target those essential genes. So the question is, does the CRISPR system that we developed work in Zymomonas mobilis, which is one of the strains that we want to use to convert sugar into things like jet fuel? And the answer is yes, with some effort. So what I'm showing you here, instead of a red protein now, it's a green protein, but other than that, totally the same sort of thing. If we have a CRISPR system where we've removed the guide RNA, so dCas9 doesn't target any genes, we get 100% green protein expression. Now, we've made a couple different versions of the Z mobilis system. Version A, we get about a 16-fold knockdown, pretty decent. Version B, we get about a 4-fold knockdown. Now, why would we want to make two different systems that have two different efficacies? Remember that essential genes are absolutely important for the viability of the cell, right? If we turn them all the way off, the cells are dead. So we can't do that. 16-fold might be too much for those essential genes. But 4-fold is right about the same amount that I'd used previously in that organism Bacillus subtilis to look for synergy between essential genes and antibiotics. So we think that this is a pretty good number right here. And we're continuing to optimize the system. We think we found the limiting factor that makes this 16-fold and not 60-fold. And we're busy poking at that at the moment. But right now, I think this is a really great place to start. All right, so let's head back to our learning goals. What did we learn? Understand the basics of genetic approaches in bacteria. Who feels like they now know some of the basics of genetic approaches in bacteria? Yeah, great, awesome. Everybody know how CRISPR works? I hear some nervous laughter. It works by using a guide RNA that matches the DNA to deliver Cas9. Now that Cas9 could be an active version of Cas9, which would cut the DNA, or it could be used like as the dCas9 that I use, which is catalytically inactive, which only represses gene expression. So it gets in the way of RNA polymerase. It's like the dimmer switch. We're just turning down the gene expression. Understand the benefits of using CRISPR to study gene function and bioenergy. No way to study those essential genes in the context of bioenergy without CRISPR, because there's no way with those classic approaches that we can actually modulate essential gene expression. Everybody feel comfortable with that one? Okay, so just really quickly to acknowledge some people, because some of this work was done when I was still a postdoc at ECSF. That's my mentor, Carol Gross. She is so awesome. She looks really awesome. She is awesome. And of course, the funding from DOE. And this is a picture of my lab, and we're doing various things, but Amy has been the real driver of engineering bacteria to produce biofuels. And so with that, I'll take any questions from anyone. Can be any question. Yes.
When you study the effect of microbes on sugar, is there any other way that we can measure the success of the microbes by looking at the output, how much output you got, and relate that to the efficiency of the process?
Yeah, that's a great question. So the question was, when we're thinking about converting microbes, converting sugar, how do we know that that process has worked? So there's really a couple ways that we do it. One is just by directly measuring the amount of sugar in the beginning of the culture before we start growing the bacteria, and then at the end. So are we using up all of the sugar? One of the other ways that we can think about doing it is simply by measuring the growth of the bacteria as well. So are the bacteria growing well in that situation? If we change the individual genes in the bacteria, do they grow better or worse? And do they change the amount of the consumption of the sugar? So yeah, we measure both the levels of the sugar and the growth of bacteria as a proxy for consumption of the sugar. Good question. Any other questions? You can ask really easy ones.
What was the citation for Lewis in 2013?
Which one was that?
I missed it. I didn't get it down.
Yes, I will go to that. If I can find it. I remember where it was. Oh, there it is.
There it is.
There it is, yeah. So this is a review article explaining about how antibiotics actually target essential gene functions. But you can imagine that these essential functions are the absolute core of the cell, right? Every single other process in the cell, no matter what the process is going to interact with these core processes, right? So if you don't understand how these core processes work, you really don't understand the function of anything. I'm not scary, I promise.
I know one of the concerns about ethanol is that once you get to a certain production level of ethanol, then the ethanol actually kills the strain itself. Is that the same case with isobutanol, and what do you do to sort of remedy that?
Yeah, so that's a great question. So the question was with thinking about ethanol toxicity to bacteria versus isobutanol toxicity and how to overcome these sorts of toxicities. So yeah, so isobutanol at some concentration will certainly impact bacterial growth, as does ethanol. The nice thing is that we're working with Zymomonas mobilis. Its normal behavior is to take sugar and convert it to ethanol. So it's used to having ethanol around. Now, we don't know if that means that it's going to be super resistant to isobutanol. We think it does have some resistance to isobutanol. But right now, actually, as we speak in the lab, someone is looking at the sensitivity of Zymomonas mobilis to isobutanol, and then looking at various genes that are involved in making the outside part of the cell and seeing if we partially turn down those genes, how does the sensitivity change? So you can imagine if we turn down a gene and it becomes more sensitive, that we could also turn up a gene and make it more resistant. And that's one of the ideas that we have by identifying some of these genes that are involved in making the bacteria more sensitive, we can actually turn that on its head by turning those genes off. Yeah, big question.
Yeah, question. So can the strain deal with both sugars from cellulose and sugars from hemicellulose, or how does that?
Yeah, honestly, I think, so the common carbon source that Xymomonas use is glucose, right? And so if we want to make it consume other sugar sources, oftentimes we have to modify the genome in other ways to do that. And I know that there are people in the center that are interested in doing that sort of thing and doing that sort of thing. And they're also working on the plant side to help change the genetics of the plant to make the sugars more degradable and more processable by these other methods. But I don't know the specifics behind that, that research. It's a good question, though.
So in the two examples that you gave with CRISPR-I, you were talking about the core essential genes and then also the cell wall genes.
Mm-hmm.
Is there kind of a broader approach you could take with CRISPR-I, something like cross between like that and transposon mutagenesis, where you could target a bunch of things, and if you're interested in a certain phenotype a microbe has, but you're not sure what genes are involved in it,
Mm-hmm.
is there a way that you could kind of target things randomly?
Yeah, so the question was, if you have, for instance, you want to understand what essential genes are doing, but you also want to understand what non-essential genes are doing, could you use both CRISPR-I and transposon mutagenesis in the same experiment? Absolutely, yeah. So remember, the CRISPR-I, all you have to do is change that 20 nucleotides. Those 20 nucleotides act as a barcode for your strain. So just by sequencing those 20 nucleotides, you know which strain has a knockdown. In transposon mutagenesis, usually what people do is they look at the sequence just outside the transposon, like 20 nucleotides outside the transposon, and they sequence that to figure out where the transposon went. So all of it is sequencing based in the end, right? So you can easily mix these two types of experiments together, both the CRISPR-I and the TN-seq, and learn about the phenotypes for all the genes. Of course, you can also just target all the genes with CRISPR-I. The disadvantage of doing that is if you don't have a strong enough level of knockdown, you won't be able to produce what they call a null, a null mutation in a non-essential gene. And what null mutation means is just that gene has no function, right? It's just, its function is totally gone. We've seen that you can approach the level of null mutations in Bacillus subtilis because the knockdown is about 150-fold, which basically means that there's nothing left, right? But for some of these other strains, we're still working on optimizing the knockdown so that we can use CRISPR-I in a way that gives us enough of a hit like TN-seq would. But yeah, and actually what we're doing with Xymomonas in the center right now is we're using a combination of CRISPR-I and TN-seq. Yeah.
Thank you so much for your presentation.
Thanks.